Good morning. Hello, everyone. Welcome. My name is Cami Mesra, and before I introduce our speaker, I have a couple business items that I'd like to address. We have 2 new members for our board next year, 2013. And the nominating committee has selected Denise Bergeron. I'm sorry if I messed that up. Denise, would you stand up? And Sonia Demirci. And I'll need the help from the audience to help us vote in these 2 ladies for our board next year. So all in favor of Sonia Demirci as our new vice president for next year, please say yay. Yay. Fantastic. All opposed, say nay. Thanks, Sonia, you may sit down. And Denise Bergeron for our current and secretary next year. All in favor, please say yay. Yay. All opposed, nay. Thank you. Thank you to Denise. Thank you to Denise and Sonia for being so gracious in accepting this position at Alice Mayo as part of the board. The Alice Mayo board is primarily run on volunteers. And without volunteers, we really can't make this happen. We also have Diana Robinson coming on as to help with our activities. And our incoming president next year will be Mariana Iglesias. I also want to thank our current board for working very hard this year in making Alice Mayo Society a success. And also on behalf of our board, I want to send a special thank you to the Alumni Society for helping in their collaborative effort of making everything we do at Alice Mayo successful. Now, without further ado, I'd like to briefly introduce Dr. Knutson. Dr. Knutson is currently an associate professor here in the Department of Immunology here at the Mayo Clinic. He has received his PhD in physiology and pharmacology in 1995 at the University of Georgia. He shortly thereafter had 2 postdoctoral PhDs fellowship, one at the University of Washington in oncology and the second in the University of British Columbia in the Department of Medicine. Dr. Knutson's passion is to find a cure for his patients. His research focuses on immunology and immunotherapy for breast and ovarian cancer. He's here today to share with us the groundbreaking research and the launch of the vaccine for breast cancer. So please help me welcome Dr. Keith Knutson.
Well, it certainly is a pleasure to be here today. So many new faces. I recognize a couple of you. If you have any questions, I think I fit the criteria of the absent-minded professor, so I may forget to cover certain things that you may recognize. So just go ahead and raise, and I'll be scanning the audience, and really that's what I like is an interactive strategy. So what I'm going to talk about today is one of the programs that, as was already mentioned, that I'm very passionate about, and that is the development of strategies to prevent breast cancer. So let me get my screen up here. I'm going to talk about work that we started about 15 years ago. And so cutting edge is maybe, somewhat problematic, but we still try to lead the way with what we're doing. And I'm going to put this all in the context of efforts that we've made both at the scientific level, but also at the national level in bringing in advocacy groups to help us push along our efforts in order to make that last leap, and that is to get it into clinical trials that are advanced stage to try to understand whether or not these approaches will work. So I'm told to do this nowadays. I have some of the technologies that I've developed here. I'm an inventor on several of them. I have 3 patents pending on the applications, and I have several licensing agreements with various companies who have provided funding for the clinical trials that I'm going to discuss, notably a company in Seattle called Tapamune, and Vaxonco in South Korea is providing funding as well. And so our efforts at development of prevention, so this is a novel strategy that we think could potentially work, and let me tell you how that's going to work, has been through the interactions of our group as well as about 4 or 5 other scientists around the world with the National Breast Cancer Coalition in what we call the Artemis Project. And I'll get to more about that in the coming slides. What is the Artemis Project? Now, I'm not here to solicit donations from the National Breast Cancer Coalition. So many of you are probably familiar with the Susan Komen Foundation or other foundations locally maybe that help breast cancer patients and drive advocacy at both the state level and the federal level. So the National Breast Cancer Coalition is probably less known. NBCC started in 1991. So this is an organization who is obviously the mission is to end breast cancer and is grassroots in that what they do is they have membership organizations of variety. It doesn't necessarily mean that they have to be named some kind of breast cancer coalition or anything like that, but small breast cancer groups from around the nation that help drive advocacy. They have a tremendous record of accomplishment over the years, and in more recent times have reestablished the need to fight breast cancer and create a sense of urgency with the development of what we call the Breast Cancer Deadline 2020. Their accomplishments are tremendous. We don't hear about them. Susan Komen, for example, it's a great organization and provides a lot of funding directly from that organization. The NBCC is responsible single-handedly for developing funding programs through our Department of Defense. Normally, these kinds of funding programs come through the NIH, but back in 1991, the President, Fran Visco, and her colleagues marched up onto Capitol Hill and said, we've got to do something about breast cancer. I'm going to talk a little bit about the epidemiology of breast cancer, but they said, we've got to do something, and we want to do it in an innovative, highly impactful way. And they requested a certain sum of money. I think it was on the order of $100 million or something like that. So they were given part of that money, and the Congress said, well, we're just going to send this over to the NIH, and they can distribute it in the ways that they would normally. And the NBC said that wasn't really what they had in mind. What they wanted to do was go outside of the box, outside of the current scientific methodology and research strategies, and go to individuals who have or were very creative and could develop novel approaches that wouldn't ordinarily be funded at the federal level by the National Institutes of Health. Well, as you can see, the program has blossomed. Breast cancer has been a solid component of the Department of Defense's funding program, which has grown tremendously to cover a wide variety of cancers, but also a wide variety of diseases. So it's a very successful program, and that's thanks to the National Breast Cancer Coalition. So back to this idea of 2020. So that seems almost unreachable. I mean, how many years is that? About 7 years from now? They started this 3 years ago. They gave themselves a decade to create that sense of urgency, to put together a solid plan that can be implemented with a variety of funding strategies to develop a cohesive group of individuals to move forward different programs that would attack metastasis, and the primary prevention of breast cancer using a number of lifestyle strategies or risk assessments, as well as a prophylactic vaccine. And that's what I'm going to talk to you about today, is that last one, that prophylactic vaccine. And what we need in doing so is the advocates that the National Breast Cancer Coalition assembles and meets regularly with, that can help us drive at the public level this concept of a prophylactic vaccine. So, we do have cancer vaccines. So you may not have thought of that. I mean, you think of influenza, you think of measles, you think of smallpox. But you don't think that we have cancer vaccines, when in fact we do. Back in the 1990s, we came up with the Hep B vaccine. And that was to prevent infection, because that was a problem too and led to hepatitis. But the long-term outcomes of Hep B infection are hepatocellular carcinoma or liver cancer. So we developed a vaccine, not we, the royal we, the scientific community, and drove that into the clinics and got it approved for the prevention of disease transmission, as well as the development of liver cancer. And in more recent times, something that's made news quite a bit is the human papillomavirus vaccine. So you probably heard of Gardasil or Cervarix. This is a vaccine that is designed to prevent transmission of the human papillomavirus, but its main feature is prevention of vaccine, and prevention of cancer, and we're seeing that now in the studies that are being done. So we have cancer vaccines. Here's the difference. We don't know of a viral cause or a microbial cause of breast cancer, so we need to take some innovative approaches to get to the point where we can test some of these novel strategies that we've been developing. Okay, so let's talk about the breast cancer, and then we'll go on into the vaccine strategies. So the normal breast consists of many different terminal ducts which are attached to these lobes, and this is where the milk is made, and of course it's transported out of the breast, and it's within these lobes where the milk is made, and here's a blow-up of that. This is where the cancer develops, and so you have these big terminal ducts here, and those you may hear the phrase ductal carcinoma in situ or ductal carcinoma. The cancer actually doesn't develop in the duct itself, the big duct here, but develops in these smaller lobule units that give rise to those ducts. And that's where we see the vast majority of the cancers developing. So we know what it looks like in terms of progression of the disease, and we can capture breast cancer fairly early now. So a normal duct that produces milk would have a single layer of cells, shown right here, and then in some individuals, for reasons that are unclear, there's the development of a multi-layered component of those single layered ducts. And this ultimately results in abnormal growth and shedding of cells into the duct. Now, many of you may know somebody that has been diagnosed with ductal carcinoma in situ, and that's a disease that's largely confined to the duct of the breast. So it's not cancer at that time point, although some people would argue with me that it is cancer. It's actually not cancer at that, because what defines cancer here is the ability of these cells to move out of the tissue. These cells that are proliferating inside here haven't moved out of the tissue yet. It's when they can start breaking down all the membranes that go around these ducts that you get cancer. So here's what we've done for cancer over the past few decades. That looks like a straight line to me. That's this is total incidence in individuals, and you can see we haven't really done much. There's a little bit of wiggling. We don't know if this decline here is something that we've done or just kind of normal distribution. And this particular graph breaks out ages as well. You can see that most of the cancers occur in older individuals, as we regularly see, but we do have a sizable component of younger women that develop breast cancer. But notice these two lines right here. This is ductal carcinoma in situ, so this is this disease here. So what happened in the 1980s that led to a strong increase in the incidence of ductal carcinoma in situ? Can anybody gather a guess? Probably mammography. That's exactly what it is. We had the ability to detect these with different types of strategies aimed at detecting lumps. And this led to an increase in the number of ductal carcinoma in situs that we detect, which is a precursor lesion of cancer. Don't get me wrong, it's not cancer yet, but it's a precursor lesion of cancer. And if you have it, you have high risk of developing breast cancer, even following removal. It's a huge problem. It's like, it's like the plague. It's like Black Death. It's 23% of all female cancers are breast cancers. And 1.5, so this is annually worldwide. The darker the color, the higher the incidence, 1.15 million new diagnoses annually, mostly in women. A few men get breast cancer. 519,000 deaths each year. Still a tremendous problem. Right now, I estimate, based on the literature that I've seen, that there's probably about 8 million people alive in the world today with a prior diagnosis of breast cancer. Overall risk, lifetime risk for a woman is 12% at developing breast cancer. So some people ask me about the role of familial breast cancer in breast cancer incidence. Only about 5 to 10% of the breast cancers can be traced through some heritable connection to family member or something like that. The rest, we have no idea. Absolutely no idea. We know that there's risk factors out there, but the average woman that comes into the clinic, I have to tell you, they don't have those risk factors. We just do not know what causes breast cancer. Okay, this is the big shock here. We're in an election period, and we're talking all about gross domestic output and deficit spending and all that. Take a look at this. This is 2006 data. 0.5% of the 2006 gross domestic product, or approximately $6.5 billion, is spent annually on breast cancer. $6.5 billion. We've got to do something about breast cancer so that we can use those resources for a better standard of living. And what this shows, and really what is the key hallmark of breast cancer, is the risk doesn't go away when you've been treated for breast cancer. And this is an example. The vast majority of breast cancers fall in this upper line here. This is a special subset of breast cancers. And this looks at the probability of being disease-free out at these various time points. And what you see is that as time moves on, this is following surgery, as time moves on, the probability of you being disease-free decreases. Look how many years that is. 20 years. You have to worry about developing breast cancer or redeveloping breast cancer 20 years after you've been treated. It would be nice, to be able to immediately treat it and have to never worry about it coming back, but that's not the case with breast cancer. For a lot of other cancers, it can be pretty much said that once you hit that 2 or 3 or 4 year mark that you're home free, and that may be the case with some. But for breast cancer, the risk still goes on and on, even with some of the novel surgical strategies that they use today. We still have patients, and on the order of about 50,000 deaths just here in the U.S. Every year due to breast cancer. So it's a major problem, and we've developed a team nationally in association with the National Breast Cancer Coalition to come up with a strategy that we could harness the power of the immune system. And I'll talk a little bit about that power. And now I'm going to get all complicated on you here. So I make it sound easy. We've got to develop a vaccine and get it into the clinic. But in fact, this is one of the hardest jobs that we can conceive of. And it's not just the scientific element, but there's the social element. There is the resources to make and package it, the distribution of it across state lines to various states so that they can administer it. And so what we're looking at and that's kind of summarized in this pipeline here where we have all of these significant challenges, is we're thinking about 3 different areas. We're thinking about bacterial and viral causes. So we're coming back to that, okay? We looked at that 20 years ago. We didn't really see much, but we didn't have the right tools. Now we're coming back to look at bacterial and viral causes. Neoantigens. Okay, so what I mean by an antigen is it's some kind of protein that's in your body that your immune system recognizes as foreign, and it sets out to attack it. A neoantigen is an antigen or a protein that's made by the tumor that the immune system says, hey, this is something that we should attack. And we'll also look at the strategies that we've been working on over the past few years, and that's the development of vaccines that target tumor-associated antigens. And don't worry about it. I'm going to come back and tell you about what those individual strategies are. So let's look at the history of vaccine. Been around for hundreds of thousands of years. In the 7th century, for example, Indian Buddhist monks drank snake venom in order to protect themselves against a snake bite. So I'm not going to go through all of these, but it was in England where this vaccination smallpox connection was made in the 1700s, which led to the development of the smallpox vaccine. We eradicated smallpox from the face of the earth with those vaccines. But we really couldn't drive the field forward as fast as we could until probably the 1970s, and up to the present time, in which now we've had revolutions in how we do science. We have all kinds of ways to look at DNA. We have all kinds of new ways to look at cells. There's just so much there that a scientist can use to study. So some of these old concepts like, does a virus cause breast cancer, we need to come back and look at those, and that's what we're going to do with some of the strategies. Okay, so what is the immune system? So that's, something that I battle every day. I'm not sure what it is, even though I'm an associate professor in immunology. It's a very complicated area. You think of it as, some system in your body that protects you against infectious diseases, and that's true. And we harness that power with vaccines, and that's shown here in smallpox, where a study measured immunity in individuals over the course of 30 years following immunization against smallpox. One of the very important things about the adaptive immune response, antibodies and T cells, and I'll come back to what those are, is they can establish memory. So you know that your brain establishes memory. Mine doesn't do so well anymore. But I bet that your immune system is just as good as your brain at establishing memory. In fact, your immune system theoretically can make on the order of 10 to the 18th connections. And that's pretty close to what the brain can do. 10 to the 18th. So 10 followed by 18 zeros. Think about how big of a number it is. That's how many memory connections your immune system can make. And we harness that power with vaccines. So we need to give you a vaccine for flu. We need to give you a vaccine for measles, et cetera, et cetera, et cetera. And that's because each one of those creates a circuit for the immune system so that it memorizes what that bug looks like, for example. And that's all contained within the blood. So you have these red blood cells in your blood, and those carry oxygen, and then you have what are called white blood cells. So some of you may have heard about white blood cells and your white blood cell count shooting through the roof or something like that when you have an infection. Those white blood cells are what constitutes the immune response. And the 3 primary types of cells that we target with the development of vaccines are what are called the B cell, the CD4 T cell, and the CD8 T cell. Okay. Now, a B cell, and I'll come back to what these are in just a moment here. The B cell produces antibodies, and antibodies are circulating proteins that bind bugs and tumor cells. This is actually a picture of a gold-coated antibody using a high-powered electron microscope. And antibodies look just like this, as we predicted, and what they do is they bind. So here's a bug. This particular one is a virus here, and you can see that there's antibodies binding. And once those antibodies bind, other cells in your body are induced to take up that bug and squash it, and kill it, and get rid of it. And that's a very effective approach at protecting against viruses. And the T cells, as I had already mentioned, they directly kill infected cells. So unlike antibodies, which are just free circulating proteins in your blood, T cells are actually small cells that have a lot of machinery that when they find a virus-infected cell, they emit all kinds of noxious substances that ultimately go to the virus-infected cell and kill it. So those are the two major subtypes of immune effectors, the T cell and the antibody, and then there's two different types of T cells which aren't important for this. But the T cells recognize viral antigens that are presented on the top of your cell. So when your upper airways get infected by flu, which I think that's coming up pretty soon now, isn't it? You're going to get infection of those cells in your bronchi or other areas of the upper respiratory tract. And your cell is going to be showing this particular peptide developed from the influenza bug. And your immune system is going to go, hey, that cell isn't appropriate because it's infected with the virus. And guess what happens? You start coughing. You start itching in your throat because your immune system is killing those cells that are infected. And that's what's happening. So when you get all the problems with coughing and everything. Okay, so why do we vaccinate? Okay, so here's why we vaccinate. Your immune system is very good at developing these circuits, these memory connections that allow it to recognize microorganisms. But here's the problem. Because it's a memory circuit, it takes a while. So that's kind of like long-term memory. You need to maybe hear it a few times so that you can get it embedded in there. It takes a week. Well, guess what? A lot of organisms, they don't require a week in order to cause infection. The coronavirus, the common cause of the flu, can do it in a couple days. And so what we do is we vaccinate in order to shrink that up. So if you were just infected with the flu virus right here, your body would take a week. And that's what it feels like when you first get the flu, is you're sick for a long period of time. Your immune system ramps up and clears the infection and then goes back down. But here's what happens at the second infection. Bam, immediately, within 24 hours, your adaptive immune response is responding to that organism again. So guess what? What do we do with vaccines? We eliminate that first infection by giving you that vaccine, right? That's your first infection. So your first infection with measles was the measles vaccine virus. That was your first infection. And indeed it was. Any measles virus that you came across subsequently would have a response against it like this. So what we want to do in breast cancer is develop a strong immune response that can immediately and rapidly and without delay attack the breast cancer to eliminate it from growing. Okay, so this is a famous 1809 painting of a soldier dying of tetanus. So this is a very important concept because your immune system can recognize very tiny doses of antigen. We want the immune system to recognize a few tumor cells, a few. We don't want it to recognize something that is a great big ball in your breast, because then it's too late, because then there's all kinds of problems with genetic instability, and your immune system may attack one part and the other part will grow out. We see it all the time in animal models. What we need to do is develop an immune response that can detect very low doses of tumor cells. And this is the same thing that we do for tetanus. We know that at the lethal dose of 50 of tetanus toxin, and what that means is the dose that kills 50 out of 100 individuals is 0.00 blah, 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 4 ounces, a lot of zeros, 1/billionth of a teaspoon. Your immune system can recognize something even smaller than that. So that's incredible. So you've got memory and you've got the ability to come in and recognize very low levels. So here's all the vaccines that we have. We have 27 different vaccines that are prevention vaccines. You see some of the favorites that we've used throughout the years. Against smallpox, that's gone now, so we don't use that vaccine. Rabies, diphtheria. Hopefully we'll get breast cancer. We've got cervical cancer there, and we've got liver cancer there. Let's get breast cancer. I think we can do it if we put our minds to it and develop a number of vaccines and test them in the clinic. Okay, so big problem. There's always a pushback about vaccines. So this is due to a paper that was published by Andrew Wakefield many, many years ago now. And you can just see from the title there where he's talking about ileal lymphoid nodular hyperplasia. Don't worry about it. I don't know what that is either. Well, anyway, nonspecific colitis. It sounds like something in the immune system. And pervasive developmental disorder in children. Guess what? This individual published using doctor data because he had a company that vaccination with the MMR vaccine, remember what the MMR vaccine is? Measles, mumps, and rubella, caused what? Autism. Oh my gosh. So what happened? The public went absolutely nuts because there was all of these cases of autism. And in fact, repeatedly it's been shown, so in this particular graph here, we show that the number or the doses of the MMR vaccine was slowly decreased over several years, and this is in a UK district, and people just stopped taking it. But guess what happened? Autism was still there. But guess what? People don't believe that data. People still think that autism is caused by vaccine. And so it's up to us as vaccinologists to move forward concepts and studies that can kill that particular perception. In fact, you can see here, this is a recent study from 2008 or 2010. I can't remember which year, but the central finding from this study was that some vaccines cause autism. 2010, folks, 1 out of 4 people still think that autism is caused by vaccines. It's not. Vaccines tend to be very, very safe. It's because of that specificity, because the immune system focuses in on what you tell it to focus in on. If it's measles vaccine, it focuses in on measles, etc. And so, and then there's been a complacency. So many of us here, I'm no exception. I mean, I didn't see polio. I didn't see, diphtheria, maybe a few cases of measles, but I didn't see any of this. So do vaccines work? I don't know. I was just given a vaccine and I never saw any of these diseases. For older generations, they saw this. They saw this impact. So diphtheria, gave a vaccine, gone. Polio, gave a vaccine, gone. Measles, gone. Guess what? The newer generations don't appreciate what vaccines have done for human health. Vaccines in combination with hygiene have saved us so much in terms of lives and dollars over the last 100 years. The biggest accomplishments in human medicine. All right. One of these days I'll get to my vaccines. So anyway, so we've got annual cases pre-vaccine of these various diseases. Looking at 2008, we can barely count many of them across the world. So not that many. So what about bacteria and viruses causing cancer? So wouldn't that be an interesting concept if we identified maybe the Epstein-Barr virus, EBV, or cytomegalovirus, CMV, or something like that, causing cancer? That would be an enormous breakthrough. That would be Nobel Prize winning material right there because of the plague that breast cancer causes in the earth. We have had so many problems trying to find bacteria or viruses in tumors because when we go in and surgically remove the tumor, all those viruses that are on the outside of your skin, guess what? They get planted right in there. So we have a problem with showing that a virus specifically causes breast cancer because there's so many viruses and bacteria in the environment that it's very, very difficult. So we know that, for example, cervical cancer, it's caused by the human papillomavirus. We knew that that was the case because we saw very strong evidence of the virus within the cancer cells. But here's the point that I wanted to make about this slide. 400 BCE, Hippocrates says cervical cancer incurable. Cervical cancer is cured. You may not see it, but it's cured. We will see within 2 decades cervical cancer completely eliminated, at least the major strains of HPV-induced cervical cancer. Okay, well that's not bad, 400 BCE to 2006. Let's think about that in terms of breast cancer. Well, we have lots of significant advances over the past couple of decades, and it took a long time getting from 1976, which was when they found the HPV, human papillomavirus DNA in cancer, in cervical cancer and warts, to 2006, where we have the first vaccine. So it's going to take a while. I'm not sure we can make it by 2020, but I'm on board with thinking about curing breast cancer, and I think that we'll have to give ourselves time, but we have to fight our way through all of the problems to get to that point. And we've been working with a number of different organizations, Lawrence Livermore Laboratories, at identifying some strategies that we can use. So there's a novel technologies. This is an example of a novel technology developed by Lawrence Livermore that we now have in the lab. Guess what this does? It detects 3,000 different microbes with, just a very small quantity of blood or tissue or something like that. We just pipette it in there with the little pipette, and we wash it away, and we see fluorescence, and we can tell what kind of microorganism. So we've done that, and we have lots of preliminary evidence that there are bacteria and viruses in breast cancer. But guess what? They look exactly like the ones that are on your skin. So we need to get rid of those using whatever strategy so we can find something that's hidden in there that can potentially be targeted with a vaccine. So as I had already mentioned, there's a number of different studies that have been done through the years. HPV, EBV, and one called MTV. These are very prevalent in the environment. These are viruses. And about 80% of us in this room carry EBV. So when I go to surgically remove a breast cancer, guess what? I'm probably going to contaminate it, even though I've done a whole bunch of, careful strategies of gloving and decontaminating, et cetera. It's a big problem, and we need to think about how we're going to handle that with respect to identifying a virus. We're not close yet. But we're still moving on. Neoantigens. So this is something that's a little bit different. Cancers make their own special proteins, and we're kind of thinking about what we can do in that regard. So this is the thread of life. So what that means is that this is the paradigm for how you get your DNA to make proteins. Proteins are what constitutes your body and what makes you rumble and roar and whatever. And so DNA encodes proteins through an intermediate called RNA. And it turns out that we now know that your DNA makes about 23,000 different proteins. Doesn't seem like much, right? We're pretty complex. We're moody, and things change from day to day, and you'd think that all those different faces of you would be take more than 23,000 proteins, but it's only 23,000. We used to think it was 100,000. However, tumor cells make their own proteins, and they do that by recombining what we call genes that are encoded by the DNA. So here's an example of the gene that encodes blue eye color, for example, and brown hair color. I'm just, making that up. We don't know that's true. But what can happen is it can take those two genes and it can make its own protein. Sometimes those proteins have very unique functions. We've isolated a lot of them from cancer. your immune system hasn't seen this before. And so we're hoping that we can develop vaccines based on what we know cancers are making. And in fact, here, Aubrey Thompson, actually in Jacksonville, recently just published a paper in which he found a whole bunch of these. So there's different kinds of breast cancer. And so we call these fusion transcripts. You know, the easiest way to think about it is, you know, just these new fused proteins here, but that's what they call it in the literature, fusion transcripts. It just sounds more scientific, right? So basically what they found was that there were 24 of these new proteins in this subset of breast cancer, 32 in this subset, et cetera, et cetera. And a lot of these were shared between the different types of cancers. So these ones that are shared, we can put into a vaccine because it's a good chance that if it works on you, it's going to work on your neighbor as well. And that's what we're looking for, something that can be universally applied to all women and not to just unique cancers. And so lastly, I want to talk about what we're doing, where we've made a lot of progress. This is where, you know, my passion has been for the last, I have to stop and count this now, probably 14 years. And I've developed several vaccines, and some of those are in large phase 3 clinical trials now. So we'll see. But we continue to improve on how we understand immunity. And so what we target in these approaches are tumor-associated antigens. So let's just say that you under normal healthy conditions, you produce these 23,000 proteins. And they're all produced at certain levels, right? Because you know that your big toe isn't producing, you know, the same level of the retinal proteins in your eye, or else you would be able to see with your big toe, right? We know that's not the case. So we know that there's a balance of proteins that are expressed throughout the body. Well, cancers tend to upregulate a lot of proteins, and at very high levels, 1,000, 100-fold, you know, compared to regular parts of the body. Your immune system sees that, and we know that because we see the immune system rolling into tumors and trying to kill it. So we've been working. We developed back in the late 1990s a series of vaccines that targeted this molecule HER2/neu. So some of you that are familiar with breast cancer know that there's a few subsets of breast cancer. HER2/neu is one of those subsets of breast cancers. It's a breast cancer that is associated with a very, very poor outcome. So patients that had this, even if they had, their, breast completely removed, they would still have a very high risk and often did die. It's a protein antigen that's overexpressed at about 1,000-fold relative to other parts of your body. And it's very antigenic, so we see immune responses being developed by all those immune cells that I was talking about, and we won't go back into what those are. And so we thought, since it's highly overexpressed, let's develop some vaccines that we can treat patients with cancer. So they've been, actually the patients have been treated with cancer and chemotherapy and surgery. They've walked out the door and they've been disease-free, okay? So they've got a good, clean bill of health. And we thought, well, let's develop vaccines that would prevent that recurrence. Because remember that one graph that I showed you that had that curve that just kept on going down, that showed that risk kept on going and going? So we wanted to prevent that recurrence. And that's the first step for us in a prevention vaccine, is to show that in these individuals. And so we developed several vaccines, and I'm not going to go into what these are. They're very complicated. We tested these in a number of early phase 1 clinical trials and had a multitude of publications on them. It was a very good experience for me because that's where I trained and that's where I had the passion for developing breast cancers. And I was never, when I moved here, I thought, well, we didn't really have a lot of data that these actually worked in patients because we were testing we were testing in early phase 1 clinical trials evaluating the safety. Well, people started using our vaccines. So they started taking those vaccines, and to make a long story shorter and to make a short story long, whichever, we shipped out these vaccines to our colleagues, Brian Zernicki at the University of Pennsylvania, And this generated immune responses. The details of this particular slide aren't important, but here's what he found. If he treated patients that walked in the door with ductal carcinoma in situ, sampled by, a biopsy. So this is biopsy where they, just take a little bit of the tissue. They can tell if it's a ductal carcinoma in situ. And he gave the patients a choice: go to surgery or take the vaccine and let's see if it works. It's a 6-month course for the vaccine. Some of the people chose, let's go for the vaccine and see if it works, because I'm interested in the development of a vaccine. 30% of the patients who this was targeted toward had complete clinical regression of their tumors when they walked in the door for their surgery 6 months later. It works. It works. And we're hoping to foster improvements in this so that we can get it into an easier form. So this particular vaccine covers only about 60%, and it's in a variety of phase 2 clinical trials now. And I'm crossing my fingers because I spent a lot of time on that vaccine. So it'll work in about 60% of individuals, so it's not a broad spectrum vaccine like we hope to get eventually, but I think it's a good first step for us.
What's the end number on that study?
What's that?
How many people were you enrolled? 20?
So this particular one is up to 47 now, if I'm not mistaken. Yeah. So these are very early trials. You can imagine that the FDA wasn't too fond of the idea of making a potential tumor wait 6 months while it was being vaccinated. But we ultimately convinced them that there was a reasonable chance that the vaccine would work. So they're more into that space now where they're more open-minded, but not completely open-minded about the idea of using a prevention vaccine for breast cancer. So Nuvax was another one of those. That sold off to a company which is now down in Texas. We did a lot of work on that. There's a huge phase 3 clinical trial going on now, which is 700 patients with HER2-positive breast cancer. The arm that that everybody hates is the placebo arm. It's very hard to enroll in these trials because you have a 50-50 chance of getting a sugar pill in place of the vaccine. But we're giving, the people that are doing this study are giving this vaccine to about 350, 400 patients. And this is just some early data from them showing vaccine versus control group. Those that got the vaccine, none of them developed recurrence over the 36-month period, whereas the control group, look at that, 25% developed it. Problem with this vaccine, it's maybe good, but it doesn't last long and it requires repeated injections, and it only works in about 35%. 35% is important, don't get me wrong. If you could prevent 35% of breast cancers from coming back, that's great. So when I moved here, we started looking at some mathematical models that we could go through these proteins again, and really, this is a big job, and this is only part of it. And using these mathematical models, we synthesized portions of that HER2 new protein, and we started looking for the development of immune responses to come up with a broader spectrum vaccine. And using some of these sophisticated strategies, we were able to see regions where the immune response really recognized as foreign. And we were able to craft those into vaccines that now will generate immunity in 90% of individuals. So that's in phase 1 clinical trial right now with here at Mayo Clinic. I'm the PI on it. We have 5 patients out of about 25. Made the FDA very nervous. Made the FDA very nervous, even though we'd tried this before. And they want me to do this in just a certain group of patients and do all of these testing things. And I think some of that's important, some of it not so much. Here's some of the important ones. You want us to do cardiac echoes. So it turns out that patients with HER2 new immunity have this antibody that they use for treatment called Herceptin. That causes heart problems. Well, they thought our vaccine, since it was related to immune system like an antibody was, would also cause heart problems, which is nonsensical, but we couldn't convince them. So they wanted me to do echocardiograms on these individuals' hearts every 2 months for 2.5 years. Can you imagine that? I'd never get that trial done. No one would enroll. Who wants to come back and do echocardiograms of the heart? So basically, you know, I battled with them, and I got them down to this many, however many that is, 7 over the course of 2.5 years. So that's more of a reasonable number, and, you know, doesn't really, you know, interested individuals that are on clinical trials that really want to help advance the science aren't too put off by this particular strategy. And so the second vaccine trial, which we've managed to open in the past, just within the past 2 months, in fact, is enrolling another 25 patients, and this one targets a new protein. This protein is expressed on 100% of ovarian cancers and about 75% of breast cancers. So I didn't tell you that HER2/neu, the one we've been working on, is only on 20% of breast cancers. So we're working on strategies that cover more patients with just a single vaccine. So it's expressed a lot on tumor cells. And we know that in breast cancer, the more of this that you have, the worse the outcome. So the faster you die. That's what's shown here. So proportion free. No, this is recurrence. Okay, so this is recurrence, and this shows that if you have expression of this protein, then out at 3 years, for example, 80% will have recurred, and if you didn't have expression, you know, you didn't have as big a chance of recurrence. So this was a good biologic molecule that seemed to play a role in the pathogenesis of breast cancer.
Where did you get the FR alpha protein? How did you isolate that?
Well, we never did. That was actually very hard to isolate. We synthesized regions of it is what we did. So we used chemical synthesizers, and we went along the stretches of the protein, and we synthesized like mimics of certain regions. And we tested, so we mapped out, so this is a protein, starts at 0 and goes to 257. That's the amount of amino acids that are in this particular protein. So we were able to use math algorithms and plot out where the immune response was hitting. So we took patients patients with ovarian and breast cancer, mapped out where the immune response was hitting. We identified the hot spots. I'm not going to go into what this is showing exactly, but you can see that patients had more or higher bars here. So those are hot spots. And we were able to formulate that into a vaccine. And I didn't bring one slide. There's one slide I really loved, which was a slide of what I had to submit. They wanted 13 copies, the FDA, and it was 1,700-page document. And, you know, that's where this pharmacology and toxicology training that I got at the University of Georgia came in handy, because they wanted me to do on 200 animals, inject the vaccine and remove all the organs from all of these animals. And I didn't want to do it, but this is what the FDA required, and show them all that data that it was going to be safe. So it was very hard. It's a very huge speed bump to get from your idea to clinical testing. You've got the FDA there. And that's good, because, you know, we see all these old movies about snake oil salesmen, and we, you know, don't really want something that's not going to work. So this one's 22 patients. In breast or ovarian cancer, patients are allowed to go into this. And the idea is, once we get this filled up, then we can open it up to advanced phase clinical trials. So I'm not going to talk about this first arrow here, but this is just a pill that they take to help the vaccine work. We vaccinated over the course monthly for 6 months, and we're gonna ask these patients to come back for a couple years. We're gonna have to take blood samples so that we can see whether or not we're generating problems in the body that may show up later, and we can see signs of that with a variety of testing strategies. So that was the easy part. So the hard part is, how do we get this into clinical testing? You know, because really the question is not, well, safety, okay, don't get me wrong, safety is very, very important, right? Then we're going to run into the Wakefield problem, where we cause all kinds of problems. No one's going to want to take the vaccine. We just spent 25 or 30 years of our life making something that we had to throw in the garbage. Safety is extremely important, but really, the next step for us is to show that these vaccines work. How we're going to do that is we're going to do it in patients that have a high risk for developing breast cancer or have high risk of recurrence of breast cancer. That's what we call efficacy testing. Does it prevent the cancer from coming back? And so those are really the next steps. So in the last few slides here, I want to talk about our understanding of the breast immune system. So this is all quite surprising data that we've collected in the past couple of, or several months, which gave some unexpected results regarding the immune system in the breast. And it turns out, just to give you the punchline, your immune system, or your mammary glands have their own immune system in there, and they look different than the rest of your immune system. So we need to think about that too. So that was kind of a little bit of a roadblock, but we're finally starting to understand, what the nature of the immune system in the breast is, and how we can modulate that with vaccines. So look, so we have all of these samples here at the Mayo Clinic from benign breast disease repository, and this is with Amy Dignam. And so what that means is I showed you all of those lesions that occur prior to cancer development. So atypical, excuse me, I gotta get this, there we go. Atypical ductal hyperplasia, proliferative disease without atypia, and non-proliferative disease. So these are all kind of lesions that occur and have some risk of breast cancer. And in fact, there's a progression here. So you can have non-proliferative thickening of these ducts. You can have funky-looking nuclei in the cells that are in those ducts, and you can have what we call atypical cells in that duct. And each of these represents a step along the pathway to developing breast cancer. So we've isolated these tissues from women that have these lesions that show up on mammography, and we've banked them, and we can see and we can follow the women, and we can say, hey, well, you have atypical ductal hyperplasia, you have a higher risk of developing breast cancer, and that's true. But what we wanted to do is try to understand, using these tissues, about the role of the immune system. And lo and behold, when we started looking at these, we didn't expect this, we saw that the immune system was clearly associated with all of these are called acini. This is where the milk is produced. This whole thing is called a lobule. But if you look at all these dots inside in the white space, that's your immune system, and that's in a normal, healthy individual. We didn't expect that. And one of the people that I work with called it lobulitis, and that makes it sound like some kind of infection. We see it in about 60–70% of normal, healthy women. And it turns out that those patients that have these precursor lesions for cancer have a lot less of those immune cells. So that's what's key. What are those immune cells doing? And we see that as you grow older, that the number of these immune cells decreases. So what are ways that we can keep these elevated if they're associated with preventing cancers from growing out? So this was all, very striking data, and we're starting to ask a lot of questions about the breast's immune system as opposed to your body's immune system. Is there a difference, and can we take advantage of that difference with the development of vaccines? And so this just shows that there are all kinds of these T cells that I told you about, CD4 and CD8 T cells in the tumor. You can see them here just kind of probing the cells that develop into cancer right here. So the cells that develop in cancer are these kind of purplish ones, and the T cells are these ones. And what they're doing is we think that they're moving inside of the breast tissue, in and out all the time, searching for cancers. We think that. And so we see that in patients that have lower levels of these cells, they have a higher risk for developing breast cancer. So that's a bonus for us if that's true, because that means that there is an active mechanism by which your immune system is preventing tumors of the breast. And how can we harness that? So what we see in individuals with these lesions that give rise to breast cancer is they have a lot less of this immune cells, like I was telling you about. Tremendously less, 3 times less. So that begs the question, can we just try to boost that up in order to prevent breast cancer? Maybe. We'll give it a shot. So with that said, I'm going to wrap it up here and talk about what we're doing in the future. We're looking at, of course, how the local immune system, as we call it, in the breast, supports the development of a prevention vaccine. Is that a normal finding? We think it is in humans. And we're still continuing to pursue the possibility of that the immune presence in the breast is due to a virus or is due to a bacteria. Because if it is, that's easy to target. We've done it with cervical cancer. We've done it with cancer, Hep B virus, et cetera. And so we think we can do it if we can identify that specific target.
Are there specific T cells in cervical cancer that's associated, when you do the histology, are there specific T cells in cervical cancer, just like in the breast?
Yes.
By the CD4
Yeah.
gene?
And that's one of the ways, so when they ultimately connected that with the virus, we have ways now where we can ask those T cells if they respond to the virus.
Do we know that much about those T cells?
About the T cells that respond to the virus? Yeah, I mean, we know that they respond to certain parts. In fact, the vaccines were based on what we knew about the immune system and how it responded to the virus. And so we used that information in order to construct the vaccine. And that's what we hope to do with breast cancer at some point. Okay, so we have lots of work to do. It's just, oh boy, it's just huge. We've got to do animal modeling. I mean, it's a night and day job, and that's why it takes a team of people, and that's why it takes the National Breast Cancer Coalition or some other organization like that. If you think I'm going to do it in my 800 square foot laboratory, probably not. Probably not. Just don't have the resources. Lots of competition. So what we need to do is hit the streets, get these guys together, associate them with the members of the advocacy community, march up to Capitol Hill, make sure that we're testing the right strategies, and get this into clinical trials across different clinics throughout the United States and worldwide. And that's how we're going to do it. And come together on putting all of the pieces in place. It's a very complex. I've been able to do it with a couple of different vaccines for early testing, so I know it can be done. But we need to get those at the national level to start doing some testing in these individuals, and I think that's where we need this tremendous teamwork that we put together. So with that said, I did this work all by myself. So we have quite a team working on the breast cancer unit, vaccine unit, that put in effort, even small increments of effort, and those that, like Amy Dagnam, who's my partner in developing breast cancer vaccines, she's a surgeon herself and is a PI on one of my trials as well, along with grant support from a wide variety of organizations. And I also like to thank Franz Visco. She's a lawyer who gave up a career to fight, starting in 1991, breast cancer, and is taking on something which has been passionate to me. She's taken this on as something that's important to her at the advocacy level. And so we hope, maybe not by 2020, but we hope within the next 10 years we'll have several vaccines that have developed because of this paradigm here that potentially we could use to prevent breast cancer. And so thank you very much for your attention. Any last questions? We can go back to lunch. Yeah.
There's a lot of those published study about perception trees patients with breast cancer. I was wondering if the team is enterprise body then, or mostly
So this team is pretty much enterprise. Well, no, it's mostly Rochester. We have people like Edith Perez is in Jacksonville, but a lot of that is local because it's easier for us to start these locally. We have a lot of significant challenges. Flying samples around by airplane is a challenge. Shipping vaccines, so we're also composition experts. We have to synthesize all these vaccines. We need to study the chemistries of them. We don't understand the problems with shipping them. So we start, at the scale where we can understand the difficulties. And the next step is to go to, 3 or 4 centers and Our discussions with the FDA, interestingly enough, they said if we're going to license one of these vaccines, we need to submit toxicity data on 29,000 people. Can you imagine how much that's going to cost? So it's pretty huge, and we have to start small. Yeah.
Speaking of costs, how has the funding from NIH been tracking up or tracking down? And what do you expect going forward?
Well, that's a politically charged question. When I came here in 2005, it was 23% at NCI. It's 6% now. 6% funding. 6 out of 100 applications get funded, whereas almost a quarter were funded when I came here. It's been dropping. We've lost about 50% of the increases that we saw over the past decade and a half. So there was a lot of money being put in. 2008 wiped out a lot of it. Do I think it's going to come back? I think we have too many problems, other problems that we need to spread the money. You know, we run a lot of deficit spending, and it depends which party gets in. So, you know, Republicans don't favor strong federal government, research programs, whereas Democrats do. So it depends, you know. It can go back up. I don't ever think that it'll go back up to the level that it was. I just don't think. So we're going to have to rely on these advocacy organizations to help drum up the money. And it's going to have to be done in a way that focuses. If the NIH doesn't focus on diseases as much as you might think they do, but with working with the advocacy community that focuses on breast cancer and getting a number of like-minded individuals, we really have an end goal in sight that we can shop around and say, you know, we have the same goal in sight. Here's what we're going to do about it. We need the money to test it.
What about endowments?
Yeah, so novel strategies. So endowments are one thing. So we've hit the pavement and we've wandered around the country trying to get small biotech companies engaged. And so we've sold about 4 or 5 of our vaccines to biotech companies over the past 6 or 7 years. So that's one way. And then they come back and say, well, we can't do the testing. We've got, you know, 8x10 storage that we operate out of, you know, over in, you know, West Palm Beach or something like that. And they come back to the clinic. So a couple times they've come back to us and said, okay, we're going to give you the money now. We've got your technology, but why don't you show us that it works? And so some of the clinical trials that you saw are through novel licensing arrangements that I've been able to craft with these companies. Yeah, that's a challenge.
It's as much of a challenge as just further the science. It seems the financial support.
Yeah, the science is easy. I mean, really, if I could just do the science, I'd be delighted, you know. But there's so many challenging things, you know. Yeah.
For tumors that don't overexpress HER2/neu, then the folate receptor alpha vaccine is appropriate, but that's the only one? That was my understanding. Is that
What's
correct?
that?
For tumors that don't overexpress the HER2 protein,
Yeah.
then the only vaccine that's appropriate is that folate receptor alpha. Is that correct?
Yeah. We overexpression is a clinical term now for HER2. So it turns out that breast cancers actually a lot of them express HER2 at different levels, and we know what that cutoff for which Herceptin is going to work. So the antibody, we know the cutoff of how much expression, and anything below that, the patient wouldn't qualify. But we think our vaccines will actually work for those that don't respond to Herceptin. We need to test it, but there's pretty good evidence that they do.
So are you going to enroll patients then who, you know, when the pathology report comes back with the receptor staining that says HER2 negative?
Yeah.
That's what I'm over.
The FDA didn't want us to do that. So I proposed that to the FDA, and they said no, because they don't understand the biology of that yet. So we need to do some more convincing with them, but that was one of the roadblocks for that. I wanted to open that up to everybody with some level of HER2 expression. They wanted it in only those high-risk individuals that were at risk for recurrence and death, and that was the patients with high levels of HER2 expression. Yeah. So that's one of the other elements is we can't always define the patient population that we want to test this in. So We'd like to, but we have to stick by rules. Okay? Thank you very much.